Welcome to the Explainer. Today, we're zooming all the way in. Way past the visible world, journeying straight into the microscopic, highly structured universe of cellular biology. If you've ever wondered how life actually organizes itself at the very most fundamental level, or, what happens when the normal rules of biology get completely thrown out the window, well, you are definitely in the right place. Let's get started. Okay, let's dive into this. Here is our roadmap for today's Explainer. We'll start with the coordinated working system, then look at prokaryotic versus eukaryotic cells, check out bacterial, plant, and animal models, dive into the cell's internal scaffolding, and finally wrap up with the acellular rule breakers. Section 1. The coordinated working system. This is the universal baseline of a cell.
So at this universal baseline, most cells operate kind of like a highly coordinated factory, and they've got three main components. Let's call them the big three. First, you have the selectively permeable plasma membrane, which basically acts as the factory gate. Second is the cytoplasm, which is this semi-fluid, jelly-like stuff that makes up the factory floor. And third, you've got a prominent nucleus functioning as the ultimate control center. Oh, and within that cytoplasm are all these tiny little organelles, this highly specialized machinery that you'd usually only be able to spot under an electron microscope.
Moving on to Section 2, Prokaryotic versus Eukaryotic cells, the great divide in biology.
Prokaryotic and eukaryotic. What do these names actually mean? Well, the secret to truly grasping this massive biological divide really just lies in the Greek roots hidden within the names themselves. It's the ultimate memory hack here.
Let's take prokaryotic first. Pro means primitive, and karyon means nucleus. So put them together, and it literally translates to a cell with a primitive nucleus. Practically speaking, this means it lacks a well-defined nucleus, and it doesn't have any membrane-bound organelles either. Basically, all its cellular activities just happen out in the open cytoplasm. It's a true open concept layout.
On the flip side, eukaryotic means true nucleus. So, unlike our open concept prokaryote, eukaryotic cells, like the ones in plants and animals, have these beautifully defined nuclei and membrane-bound organelles. They operate with specialized, totally enclosed departments for specific tasks. Everything has its own little room.
Now, what's really interesting about this slide is when you place these domains side by side, the contrast is just stark. Look at the scale here. A typical prokaryotic cell is just 1 to 10 micrometers in diameter. Tiny. Eukaryotes, on the other hand, are absolutely massive by comparison. We're talking 10 to 100 micrometers. And while prokaryotes usually operate as these solitary unicellular creatures, eukaryotes are what build the vastly complex multicellular organisms we see all around us every single day.
Section 3. Bacterial, plant, and animal. Let's look at 3 typical cell models. All right, looking over on the left, you can see the bacterial cell operates without a distinct circular nucleus. Its genetic material is just kind of bundled up there in a nucleoid region. But scan your eyes over to the plant and animal cells. Both of these are eukaryotic, and right away you instantly see the prominent, well-defined nuclei and all those specialized structures, like the massive central vacuole and chloroplasts in the plant cell and the dense mitochondria packed into the animal cell. The jump in complexity is just visual and totally immediate.
Based on those architectures we just looked at, mapping out these features actually becomes a pretty simple exercise in logic. Try filling this out in your head. The exact moment you identify a primitive nucleoid, you know for sure you're looking at a bacterial cell. Whereas, the presence of true, membrane-bound organelles instantly flags the complex structure of a plant or animal cell. It's like a biological cheat code.
All right, Section 4. The cell's internal scaffolding. These are the support systems inside the eukaryote.
The cytoplasm is far from just empty space. It actually houses 2 completely distinct hidden dynamics. And this brilliantly illustrates how cells work. On one hand, you have the active cytoskeleton, which is this microscopic network of really fine fibers that provide support, shape, and handles internal transport. On the other hand, you have passive cell inclusions. Think of these as the cytoplasm storage vaults. They hold everything from starch to literal crystals of calcium oxalate. It's just an incredibly efficient use of microscopic real estate.
Which brings us to Section 5, Acellular, the rule breakers. Entities that totally challenge the definition of life.
Now, acellular agents completely lack the cellular machinery we've just spent all this time mapping out. No plasma membrane, no cytoplasm, no organelles, nothing.
It's actually a fascinating progression of just stripping away parts. First, you have viruses, which are just genetic material wrapped up in a protein coat. Next, viroids, which is literally just the genetic material, no protein coat at all. And finally, prions, which are just misfolded proteins with absolutely zero genetic material. Yet somehow, all three are highly infectious agents capable of hijacking those complex eukaryotic cells we just talked about. Wild, right?
And that brings us to a really profound biological paradox. If viruses, viroids, and prions completely lack cellular structure, if they are completely missing the membrane, the cytoplasm, the nucleus, but they still act alive, multiplying and infecting hosts, where do we truly draw the boundary of life? Is life strictly a cellular phenomenon? Or is there some shadow biosphere out there operating by entirely different rules? It's a question that challenges the very foundation of biology itself. Think about that for a bit. Thanks so much for joining me on this explainer today. Keep questioning everything, and I'll see you next time.